Hello, and thank you for joining today's cutting-edge conversation about the challenges and innovations in CMC, chemistry, manufacturing, and controls. We appreciate you joining and looking forward to having a lively discussion among our esteemed panel and with you, our audience. I will begin today's webinar by introducing you to our speakers. I'll kick it off with a high-level overview of the challenges and solutions we have developed in the CMC space, and then you'll hear a presentation from each of our panelists about a particular approach or technology that offers a proposed solution to the various challenges surrounding the CMC area. It is heavy on a practical application, but rest assured, these speakers and their companies have enormous expertise and diverse teams that support their subject matter knowledge. I would love to hear you throughout this presentation. Submit your questions by using the Q&A function, which you should see at the bottom of your Zoom window with the large letters Q&A. If you are interested in getting in touch with any of our speakers after the webinar, their contact information will be available on the last slide, and you can access each organization through the Scientistcom marketplace. Now I'll begin with introductions. My name is Frederik de Rose, and I will be your host today. I'm Vice President at Scientistcom, leading the chemistry category, including CMC. I've been in the pharma industry for 30 plus years, including Johnson & Johnson, Janssen Pharmaceutica, for 20 years, mainly in oncology and anti-infectives. I've been involved in 15 plus investigational new drug applications and submissions. Let's meet our speakers and panelists. First, we have Dr. Hulger von Müller from moloX. Hulger studied biochemistry in Berlin and Bochum, did his PhD at EMBL Heidelberg, then worked as a postdoc at MPI Martensried, and moved with an EMBO fellowship to the Free University of Berlin. He co-founded moloX in 2012, and he's the CEO since then. He has 20 years of experience in proteins and protein crystallography. As second speaker, we have Fernando Huerta from RISE Research Institutes of Sweden. Fernando Huerta received his BSc and PhD degree in organic chemistry from the University of Alicante. After a postdoctoral period at Stockholm University, working with dynamic kinetic resolution of secondary alcohols, he took an associate professor position at the University of Alicante. In 2002, he moved to Stockholm, where he started his career as a medicinal chemist at AstraZeneca. Together with two former AstraZeneca colleagues, Fernando founded Chemnotia in Stockholm in 2012. Fernando was a key player in the development of IC forward reaction prediction. Since 2016, Fernando has worked as a senior researcher project leader at the Research Institutes of Sweden, RISE. And then last but not least, we have Dr. Rahuman, who is the Senior Vice President of and Head Chemical Development Solutions at Aragen. Dr. Rahuman has 2 decades of expertise in handling NCE process development, PRAMs, generic API development, DMF filing, technology development, innovation, and cGMP manufacturing projects. He was responsible for the design and commissioning of the Aragen Kilo Lab, process development lab, and pilot plant. Prior to Aragen, he worked with Syngene International, Dr. Reddy's Laboratories, Shazun Research Center, and SABIC Technology Center. So thank you, panelists, and thank you, attendees, for joining today. I will now begin with introducing the current challenges surrounding sourcing CMC. The problem. Sourcing CMC is difficult and time-consuming, as I'm sure you all know. We all know that there are thousands of CMC suppliers globally, and we at Scientistcom have learned during the global pandemic, there is a high need for local and regional suppliers. It's difficult to identify suppliers, maintain information on certifications, capacity, capabilities, and ratings. It is challenging for every research organization to ensure compliance to policies and procedures in a highly regulated space such as CMC. Last but not least, it is not that easy to find and source innovative CMC tools and technologies such as analytics, formulation, etc., for new therapies being developed for both small and large molecules and anything in between. What we have done is enable our clients to have the diverse supplier base and transparency. They can still have their own core strategic suppliers. They can access them through the Scientist.com platform, but they can quickly access additional suppliers as they require. We have consolidated supplier information into a single location to support our clients with their supplier management. We have addressed the compliance requirements in advance, and we are looking into giving our clients additional functionalities for further guidance and control requests and control research behaviors. This is how the outsourcing landscape looked before Scientist.com. And this is how the outsourcing landscape looked after and with Scientist.com. So one global supplier base, which improves quality, saves you time and money, gives you access to innovation, and increases compliance in every step of the process. Most of you do have your own Scientist.com enabled marketplace, I'm sure. If you would not have it already in place, please contact me at any time. And on that note, I would like now to pass this to our first panelist, who will cover Every Crystal Counts. It's my pleasure to introduce our first speaker, Dr. Hulger von Muller from moloX. Hulger, over to you.
Hello, can you hear me? Good. So thank you, Frederick, for the nice introduction, and thank you for the opportunity to present moloX here today. I will quickly introduce the company, who we are, where we come from, and then I will talk a bit about structural biology and what we can do today with crystallography, and that this method can be used as a screening method. That's our next slide. So moloX was founded by myself and Bernard Loll. This is also where our name comes from. This is about 9 years ago. We are currently 5 scientists. We have no third-party funding, so we are independent. We have clients from big pharma to chemical industry to biotech and worldwide. And we have partnerships and contracts with academic institutions, as well as synchrotrons and other leading CROs in the field. Next slide, please. So moloX was originally founded to produce proteins for academic labs. This is we wanted to make proteins for, especially for the RNA protein world, because we thought commercially available proteins were not good enough and too expensive. And so when we founded the company, we were immediately approached by a big pharma player, because we are all crystallographers, and they asked us if we could help out with data collection, so X-ray diffraction data collection. And so we shifted gears and never came back to the original idea. Today, our revenue comes from partially from structural biology, so data collection, as well as crystallography and EM, but also the other side is protein purification on demand. And next slide. I will not talk about the protein parts today, but only about structural biology. And here very quickly, moloX is offering two sides, complementary structural biology methods. We're offering cryo-EM for about 3 years now successfully. It's a great technique that is developing very fast at the moment. It has a lot of bottlenecks still. It's very tedious and time consuming, and has a lot of bottlenecks in general, but it's developing technically very fast. So we are expecting a lot from this, but I'm not talking about this today. You can ask me about this. I'm happy to give you an insight into this. Crystallography, on the other hand, is known for decades. It's used in industry for decades as well. But as in cryo-EM, the technical development has pushed the limits within the last 5 years, especially. And this is especially true because the synchrotrons and the beamlines improved so much, and especially the detectors improved so much, so that the overall throughput that can be generated has accelerated everything. At moloX, we can offer all steps in crystallography, so from crystallization to data collection. You can send us your crystals only, and we collect data on them. We are among the leading data collection service providers here worldwide. We have contracts with all relevant European synchrotrons. We can help with data processing, structure determination, refinement, model building, and so on. We also help clients with data management and software consulting. So if you have questions about your pipelines, we are happy to help. And more recently, we established a collaboration with a company called Nuvisan to do large-scale ligand screening campaigns. And I want to go back a little bit to the development at synchrotrons. So when I started my PhD, the typical time for a typical data set was 20 to 30 minutes. And today we're talking about 20 to 30 seconds for the same or even fourfold the amount of data. And next slide. I will now introduce you to this collaboration that we started with 3 players involved, actually 4 players involved, because there's also a client, a big pharma client in the background. We have us and Nuvisan ICB. Nuvisan ICB is a newly founded company this year here in Berlin. They bought parts of Bayer Research and are now more than 400 employees. And they offer all kinds of integrated services in the drug discovery pathway. And since they have the facilities and also the manpower to handle these large ligand campaigns, and we have the manpower and knowledge to do the data collection for this, we joined forces. And we also have an academic partner here, EMBL, that run the beamlines we use. And I will quickly guide you through the slide here. Crystallography has been automated for a decade now or more. So people used robotics to set up crystallization trays for a very long time. So if you have a crystallization system, making crystals is usually not a bottleneck anymore. And what is new now that if you go down the road, if you have 1,000 ligands or even bigger campaigns, you need powerful techniques to so-called soak the crystals with your ligands. And at Nuvisan, they have a sonic dispensing system called ECHO that can handle all kinds of ligands from 96 well to 384 wells, or even smaller miniaturized formats. And from there, it can shoot by sonic dispensing ligands to a target. And in our case, this is a crystal. So this is very efficient because it uses very little material, and it's super fast. And it doesn't really matter if you do 2 ligands or 2,000 ligands, the machine can handle that super fast. At Nuvisan, they also overcome a bottleneck, which is crystal harvesting, because they use a semi-automated system called Shifter where the technicians now can freeze 50 to 60 crystals per hour, and that includes packing them in dry shippers to be ready to be shipped to the synchrotrons. And so a typical day, you can manage hundreds of crystals here. We then joined forces with EMBL to introduce and develop a fast semi-automated data collection platform to increase the speed and throughput, and at the same time be able to react to difficult samples, because from a lot of automation processes, we see that difficult samples are just lost in this pathway, and we believe, especially because the samples are getting more and more expensive because they come from, for example, mammalian expression systems, and the amount of work that is put into these samples to generate them is way more expensive than losing one data set and spending the time here to adapt to very difficult crystals and collect the best possible data from each one of them. And then down the road, there's automation on the data processing line is already implemented in crystallography a long time ago, same as ligand finding, ligand docking, and so on. The ligand part, I can't tell you much about this because this is the client's work in this case. And but yeah. So overall, I can tell you here that we did this in lightning speed. 200 ligands took us 2 weeks to get from protein that was delivered from the client to the raw diffraction data, and then the client decided to do another 1,000 ligands on top. And although we had a summer vacation time, this was done in 6 weeks. So next slide, please. I want to sum this up now a little bit. So I showed you here that crystallography can be used as a semi-automated ligand screening method, and it's feasible and very fast. It's very efficient and cost effective. And I already showed you the time frame here. It can be done in very fast time frames, especially because Nuvisan and us together have more than 10 crystallographers that can handle also the downstream work. I want to tell you a little bit about the success rate, and I think here we are probably better than others. Because we have this semi-automated but still human-operated system, where from 1,200 ligands we got 1,071 data sets, with 97% of those are below 2.3 angstrom resolution, which is our cutoff for automated ligand finding pipeline. And the median resolution here is 1.84 angstrom with fantastic R factors. And with this, I want to just emphasize that you should consider implementing these screening methods very early in your drug discovery pathway, as well as in late stages. And this is not only limited to fragments or small molecules. This could also be DNA or RNA or peptide binders or even other things. And prices in general, I can say Beamline prices in the last 10 years have not changed, but the throughput has dramatically changed. So the per data set price is very good at the moment. So we can do these screening campaigns by crystallography now. And with this, I would like to thank everyone involved, especially people from Nuvisan, Martina Schäfer and Tamo Stevens. People from EMBL, Thomas Schneider's group, especially Greg Burenkov, and from my lab, it's Sophie Zimmermann and Christian Becker. Thank you.
Thank you so much, Hulger. It was a great overview of why every crystal matters. Thank you so much.
I hope I was on time.
Absolutely, no worries. Very good. So our next speaker, let's move to the next speaker, is Dr. Fernando Huerta. Over to you, Fernando.
Thank you, Frederick, for a nice introduction, and thank you everyone for putting this together. I think it's a great thing to do in these days. And before I start with my scientific talk about retrosynthetic analysis, let me talk a little bit about what is RISE. If you go to the next slide, please. Well, I guess you can read much faster than I can speak. So I would just say that RISE is the result of a great idea from the Swedish government, putting together more than 30 different research institutes to work together. And then in this way, we can tackle all interdisciplinary problems nowadays. So I think it was absolutely great, this idea, and we are one part of it. Next slide. Within the Department of Chemical Process and Pharmaceutical Development, well, we can take care of small pharmaceutical development projects up to take the whole preclinical development of a drug candidate and also GMP manufacture for clinical trials. If you have more questions about this, Håkan will be very, very pleased to answer all these questions. So let's go to the scientific presentation. Next slide, please. Well, I'm going to talk about retrosynthetic analysis and take a stroll on memory lane. But before of this, I want to say that retrosynthetic analysis is very old topic, is huge. Just to show you how old it is, in the next slide, you can see some of the first molecules to be synthesized. I mean, the most famous one is the urea that was synthesized by Waller back in 1828. It took almost 20 years before one more carbon atom was added to the synthesis, and in 1845, Kolbe synthesized acetic acid. Still in the 19th century, Hoffman synthesized acetyl salicylic acid. Next slide. I mean, we go back to the title, retrosynthetic analysis, stroll on memory lane. I mean, obviously, this has to be a very quick stroll because we have not much time. So let me jump in time to back in the mid 20th century. At that time, there was a debate. And I think you are going to allow me to bring this debate back nowadays. In the next slide, we can see these two great chemists, Nobel Prize winners in synthetic chemistry, Robert Woodward and Alexander Todd. They said back in time that organic synthesis was an art. On the other side of this debate table, next slide, we have Elias James Corey. I think it was back in 1969, I recall that he wrote something like, soon computers will design and plan synthesis better and faster than humans. I mean, this is an impossible debate. We cannot solve this now, but just let me show you in the next slide what is another definition of art, what they mean, what actually they meant. Look at the complexity of the molecules they were synthesizing back in time. I mean, Woodward synthesized vitamin B12. Yeah, there is no more to say about this. If you ask me, I will buy this as another definition of art. But what about computers? In the next slide, I put together some of the actual systems, the software we have nowadays to help us with retrosynthetic analysis. There are many more. Yes, I put a selection here. I would like to remark just two of them. ARChem, now it's called ChemPlanner, and ICSynth. I was deeply involved in the development of ICSynth, and very, very lucky to be one of the first ones trying ARChem in the early days. Another mention on the bottom of this list, we have ISIS-Finder and ASCOS. These are two open source systems that they work, or the back engine is using machine learning algorithms. So if we go to the next slide, we have to ask a question. I mean, when we do retrosynthetic analysis, this is art or this is science. I mean, it's very difficult at this stage to decide which, what we're going to do. So let me introduce in the next slide what is the main character of this presentation. Of course, if we are talking about synthesis, we have to talk about target molecule, otherwise there is no possibility. So what we are asking ourselves if we are going to do analysis of this molecule, next slide, are we going to use traditional approach or modern methods? Well, just to decide which one, we have to make at this moment a break, parenthesis, and see what is the state of the art. Next slide. When I was preparing this, I was thinking how to explain this, you know, and there are many, many different possibilities. We can talk about the different algorithms, the neural networks, the natural language processes that they are used nowadays to handle chemistry. But I think it's faster to explain the state of the art with a metaphor. So in the next slide, I found this, this legend, and I thought it was quite interesting. I mean, all roads lead to Rome. And I was thinking, okay, if you take Rome as, you know, your target and all the roads, just the different synthetic pathways, and we analyze how this has evolved over the time, just thinking how our pioneers at the beginning were doing this synthesis. In the next slide, we can see that probably they were using a Fiat 500. They had to drive themselves. They didn't have any electronics. They need to know all the, you know, different streets and ways to get to the destination. Compared with what we have nowadays, next slide, I would say nowadays we are driving a medium-sized SUV. We have GPS, we have our telephones that tell us if there is an accident or so, but still we have to drive ourselves. And we will find problems along the way. What is the future? Next slide. Well, the future, as I see it, is when you just put the destination and let the car drive yourself. Unfortunately, we are quite far away from this situation. So I'm going to show you what is the ugly truth about the retrosynthetic analysis nowadays. Next slide. And if we are going to do an analysis for this particular molecule, and the first things you do is, next slide, you go to your experts. And then you do, you know, these fancy words like we have 100 years experience in chemistry or brainstorms or whatever. At the end of the day, your experts are going to recognize that bond in between the two indole moieties as the most logical bond to disconnect. Once you have this idea, you go to SciFinder or any other reaction database, and you look for any example that is similar. And here you go. You have one example in SciFinder that is quite similar. So we are very good. If, you know, this task is given to you by an important customer, you want to do something else. So you go to the CAS, the computer-assisted synthesis design systems that they are out there, and you try on them. I mean, we use here, the first one is Chem Planner. And you see, I'm marking with red dots. You see that they gives you the same disconnection. Okay, let's try something else. ISIS Finder. Okay, the same disconnection. ASCOS. And you find exactly the same disconnection. And guys, listen to me. This is terrific. I remember the time when a computer couldn't even map a reaction. So the fact that now we have systems that can mimic what our expert says is great. But wait a second, what we do now? We got this task, we do the analysis, we, you know, brainstorms, experts, blah, blah, blah. And now what we do, we write a report and we go back to our customers, say, look, we have done this and that, and we found that this is the best, not the best, it's the only solution we found. So for me, the next question is obvious. Next slide. Is that it? Are we going to be satisfied with this? So let me show you how we do in RISE. Next slide, please. We have a method how to do this kind of retrosynthetic analysis. And we use all the tools we have in our toolbox for synthetic analysis. So next slide. When we get this kind of task, what we do is to look for transformations. We look for transformations that allowed us to make all bonds that we can disconnect with a synthetic logic. What I mean with this is that obviously we are not going to disconnect the bond between the 6 and the 5 member break in the indole. That's not logic. Here is just a representation of 6 disconnection. We look for much more of them. Furthermore, we don't use just substructure or structure. We use graph, in this case, a subgraph of the target molecule. And the goal is that we want to find all transformations within the network of organic reactions. If we analyze, one of these disconnections, we don't care which substituents they are around the indole. So we don't care if the nitrogen is substituted by another heteroatom. We do that analysis afterwards, but we want to find it all. Once we do all this process and it's done in a semi-automatic way, next slide, we are able to come up with more suggestions than just using traditional approach or the computer approach. You can see here, synthetic alternative 1 is the experts approach, but we were able to find another one. Just summarize, next slide. I would say that we have a wider coverage of the network of organic reactions. We have in-house tools to help us to take the right decision in which synthesis is more suitable. Quite often, we find out-of-the-box synthetic solutions. And let me explain. Sometimes out of the box doesn't mean they are the best. But anyway, you have to find them if your customer wants to patent or wants to cover everything. So I would say that we use a combination of traditional and modern retrosynthetic analysis. But if we go back to the origin, I mean, retrosynthetic analysis, art of science, next slide. I think we use both, but we were not the first ones. Thank you.
Thank you so much, Fernando. It's a really nice presentation. I think it's a map of Paris. Huh? Anyway, we'll come back to that. But a really nice presentation. Can I just remind everybody, the attendees, that you can send your questions in the Q&A box, so don't forget to do that. I see there is already a question, so thanks for that. So now we will go to the third panelist, which will cover how to accelerate IND submission, GMP production. It's my pleasure to introduce you to Dr. Rahuman from Aragen. Over to you.
Thank you, Frederick, for your kind words. I think I'll be covering on CMC accelerating IND submission. Again, as a part of that, I'll be covering about the GMP production and how Aragen can support the drug discovery and development in this area of CMC filing. As most of you might be knowing, Aragen is one of the leading CRO located in India. We are based in 4 locations in India. And of course, our biologics unit is located in US. So I'll be covering as part of my presentation about some kind of an introduction about what we are and what we are doing. And you can move on to next page, Frederick. So our purpose and belief in this CRO domain is we definitely strongly believe that in every molecule is a possibility for better health. Our purpose motivates us to drive the success of your programs so that we can together transform hope into health for millions of people around the world. As you know that, I think most of the unmet medical needs, we need a lot of discovery and development program, and where we can partner and collaborate with you and try to find a solution for the unmet medical needs. You can move on to next page, Frederick. So to give a flavor about Aragen, we are closely 2,800 plus scientists. Apart in that scientist population, we have closely 400 plus PhDs and postdocs. And it's a very vibrant talent pool located predominantly in Bangalore and Vizag, and of course in Hyderabad in India. These are the places where we will get abundant amount of talent pools from various universities and institutes. And we support at this moment of time 450 plus customers. On top of that, we from the top 10 pharma, we support 7 pharma companies and 100 plus biotech companies for various discovery and development and formulation and of course biology programs. Every year, typically we run 175 plus programs. And out of that, in the development domain, we work typically 70 to 75 projects and 15 plus of late phase development activities, predominantly early phase and all the way to IND filing, phase 1, phase 2. And of course, 8 to 10 molecule we also support for commercial launch and phase 3. Kind of industries we serve, of course, as I mentioned, is a CRO, not only for pharma and biotech, we also support for animal health, agrochemical, nutrition, and performance chemical. So this is our scientific pool available for the contract research organization. And my next slide, I'll be talking about the advantage of going with Aragen and what Aragen integrated drug discovery and development platform can offer. As you know that there are new CROs available across the world, how Aragen is differentiating between CROs and the main territories. We have all kind of arrangements like starting from discovery and also development, pre-formulation, formulation, biologics, biology. All these activities can happen at one place. That all is a rapid speed time to clinic. Rapid means it is research, pharmaceutical development. Some of programs, when we start from the discovery all the way to IND filing, typically it takes 5 to 6 years if you start approaching multiple companies for every single activities. Say it is discovering your own hit identification or like doing a pre-formulation. When you approach different industries who are expertise on different domain, overall timeline it is going to take more than 5 to 6 years. That's a typical industry now. However, in Aragen, since we have all these verticals are like work together seamlessly, from the hit identification all the way to like GLP talks and IND filing, we could manage like at least 43 months time. That amount into almost 32% of time saving and advantage which you are getting it by approaching Aragen. Here we have a large scientific pools and they work on hit identification. So since we do this activity seamlessly in Aragen, we are able to save some amount of time. Now, going forward, I will be giving few examples how our like discovery programs are taken to all the way to GLP talks and IND filing in a faster mode. You can move on to the next slide, Frederick. Yeah, the next slide. Yeah, so I'm just giving a case studies. My first study is on the development of an NCE molecule. The molecule was developed from our Medcom team. Internally, it got developed. So the molecule which got developed on the milligram to gram scale, it came to the development team within Aragen. When it came, it was around 17-step synthesis. The task given to us is like to produce closely between 2 to 2.5 kilo of non-GMP material for toxicological studies and a kilo of material for phase 1 studies. So we could learn whatever is already done from the Medcom scientists, and we did not waste a lot of time, and we could quickly turn around the process and identify that what are the places we can shortcut, we can do a telescoping, or we can like synthesize and reduce some number of steps. And we could manage the 17-step into 14-step synthesis. Typically in the Medcom, we used to do a lot of column purification, and which we could avoid in the development stage, and we could do with one filter column. And overall yield also we could improve in a shorter span of time. Usually when the 17-step synthesis, it is a linear, it was giving a less than a percentage of yield, and we could improve in a shorter duration of time all the way to 6%. The outcome, we could support the non-GMP material 7 weeks ahead of time. And of course, the GMP also got reduced 10 weeks. So overall, it is almost a 17 weeks time which is saving, which is closely 4 months time if these things are happening in parallel in two different organizations, it may not be possible to save such kind of time. And at the end, like we are able to support the IND filing. And currently we are in the process of supplying the phase 2 material. My next case study is, Frederick, you can move on to the next slide. My next case study is again a very interesting molecule which was identified within Aragen from the Medcom team. Initially, it was a 7-step synthesis, and we supported the tox and GMP material. When it was given to us, we quickly took a scanned the molecule with an alternative strategy. Initially, we came out with a 6-step synthesis. We could reduce 1 and supplied the material for tox studies. When we supplied the material to tox studies, we continuously worked on the molecule and see how quickly we can reduce the number of stages and the turnaround time. And our scientists to come out with 3-step synthesis with completely alternative route. And with that, we are able to increase the yield from less than 18 to 40% yield. And the overall cycle time for a 6-step process, typically you start from the first stage all the way to 6 stages, typically take 40 to 45 days per batch to reach all the way to the API with the desired form. Now we are able to make it within 20 days time. So definitely we could save closely 13 weeks time, which is a great help for our customers. And they are in the process of moving ahead with phase 2 supply. Again, we are contributing tremendously and supplying more than 250 kilo of GMP material for their phase 1 studies, and also right now working on phase 2. Apart, you can move on to next page, Frederick. Apart from supplying the NCE molecule, we also support some of the advanced intermediate for NCE molecule. For example, a carbohydrate chemistry where 8-step synthesis we started working as a development project. Numerous amount of column purification was there. When we got the Medcom process, we changed everything, and we could supply 9.5 kilo instead of original target of 5 kilo. And with that, we could collaborate with another innovator, and this innovator is from Europe, and we are able to supply GMP grade intermediate for the NCE development. My next example is again the similar way, again very advanced intermediate for an NCE molecule, and we are the supplier for even this intermediate, where we are able to develop a robust process up to 700 kilo GMP material, and definitely this has helped the innovators to accelerate their molecule to go to clinics and phase 3. You can move on to the next page, Frederick. Now, even though a couple of challenges, whatever you face, we have an advantage in Aragen, like how can we go faster? That might be the question as an innovator or innovative virtual biotech company. Coming to Aragen, since we use the integrated approach, you will be able to save substantial amount of time from the discovery all the way to pre-formulation, formulation, and biological studies, and of course development. And this activity can typically happen seamlessly, so that you don't need to transfer the technology from one organization to another organization. So that's an advantage. By coming to Aragen, you are going to get it. How can I quickly get access to relevant information? We have a very robust customer and centric communications. Entire communications are managed by PMB certified professionals, and they connect internally with all the cross-functional team. And also we use modern tools, electronic lab notebook, and various databases. And we don't use typical emails and for sending them updates. Everything is through share folder or various other technology platform we use, so that the intellectual property is well maintained. There is no leak of information. And our track record is we have more than 1,400 synthetic chemists, 300 process chemists, and we, as a part of this entire endeavor, we have developed more than 25 plus formulation. And on an average per year, We support 10 plus IND program and 70 plus clinical program per year average we support. And almost like 50% of our customer works with either discovery and development or development and formulation or discovery and biology. More than one vertical they work. And the biggest plus point is we have a large pool of scientists available in the discovery and midcom. So on need arises, we will be able to utilize the resources, and we are scattered around 7 sites. So we don't need to worry about like if there are a site, we have some challenges due to local issues or local epidemic issues. Our scientists are scattered in multiple locations. We'll be able to manage your program seamlessly. And we have a good track record on regulatory and excellent facility, GMP production facilities. We have more than 6 clean rooms. At a time, we'll be able to manage 6 final material API, and more than 10 intermediate will be able to manage. That's all pretty much on CMC, and like you can move on to the next page. We collaborate. Our motive is your science, our solution. Together, we can make success and meet the unmet medical needs. Thank you. And with that, I would like to take any questions if you have. Thank you.
Thank you so much, Dr. Rahman. And it's glad that you were had only a very small technical interruption. No worries.
Yeah.
I will go straight away into the Q&A session because we only have limited time left, if you don't mind. The first question is actually for you, Hulger, on somebody was asking, hope your screening covers the GPCR target structures as well. I don't know if you can comment, please, Hulger, if you can.
Yes, so in general, I'm happy to discuss this in person, but it's GPCRs are known to give very small crystals, so they have to have a certain size in order to be able to collect the full data set on one crystal. Otherwise, you lose throughput, let's say. And even if you collect multiple data sets from multiple crystals, this is doable, but this is probably not high throughput anymore. But this goes in the same direction as I mentioned before. The sample is so precious and so expensive that it's worthwhile to get the best data out of these.
Okay, thank you, Hulger. I'm just gonna jump to the next question. This is actually for you, Fernando. On somebody was also asking, indeed. So once you have all these new routes, theoretical routes, are these routes actually explored, tested again in the lab for feasibility? So what are the next steps? If you can comment a bit on that, to the wider audience, because I saw you already responded a little bit on the chat.
Yes,
maybe you can just repeat.
Yeah, thank you, Frederick. The answer is yes. I mean, of course, when you do this kind of analysis, you have to be to have a very good communication with the customer. The main reason is because they know the chemistry better than we do. So even we can come up with solutions, they know which step or they know which route they prefer. So we present all our findings to our customers, and together we take a decision which is the more suitable for them and has more chance of success. So, but yes, the answer is yes, of course. We go to the lab and try them.
Thank you. A little bit related when I'm just where we are with you, Fernando, is another question that just came in is, what is the best way to look out of the box, as you say, the out of the box approach for synthesis of new molecules? What is actually your recommendation?
Oh, this is a very tricky,
yeah, I know.
It's a very tricky one. I mean, nowadays there are so many different alternatives. I mean, you can use, of course, experts in chemistry, all the innovative software we have out there. My recommendation is use everything you have in your toolbox, and don't focus just on computers or what your experts and your knowledge in chemistry says. Just look for all kinds of alternatives, and the method that we have help us, because sometimes when you disconnect a bond, you don't know if it's possible to connect it, but we look for that chemistry even if we don't know that exists. And that's the way. Just try and error. Sorry, there is no better answer than that.
All right. No, thank you. Thank you very much for that. Before I go to Aragen, I have another question actually for you, Hulger. So what is the difference to other fragment screening providers? Example, the diamond synchrotron. Can you maybe comment a bit on the difference?
In general, I think I don't want to give numbers here, but I think the throughput with us is much higher than any automated beamline. Plus, as I said before, we don't want to lose any precious samples on the way. And I know that people think that high throughput, you have a fraction of samples that you just lose by chance or by mistake or whatever. But I don't think this is necessary, and I think you should always go back and look at these more difficult samples or not lose them in the first place. And then I think this is a big difference: speed, which equals price, and that we make sure that you get the best data out of each possible sample.
Thank you, Holger. I have a question actually for Dr. Rahuman. I mean, there's a lot of questions, but I think some of them can also be answered offline. But maybe just to starting with your success rate, if you can comment, because you mentioned that you have done a lot of IND filings. Can you comment a bit on your success rate for IND filing? Yeah, so typically in a year, we do around 5 to 6 programs, we do it. So I can say success rate is around 25% at this moment of time. Hello. Yeah, sure. I was muted. Yeah. Thank you very much. Around 25%, I can say that moves forward. And wherever we have some challenges, maybe the molecule comes back and we need to do further some more studies, and we go back. And I mean, also a little bit a tricky question that just came in also for you, Rahman, is what is it? I don't know if you can say, but what is the typical time for a first-in-human batch for about, let's say, a 6 to 8 step synthesis? It's maybe a tricky one with so little information, but can you comment a bit in general? I definitely can comment, Frederick. So if we have done the tox material by ourselves, for example, if the tox material is done by us, then the first-in-human is going to be very straightforward. For 6-step synthesis, typically we take 3 to 4 months time. However, if we need to do only the first-in-human, if the tox is done somewhere else, and then we may take additional 2 more months. Reason being, we need to go for feasibility, and because our documentation require proper consistency batch before we go for a GMB. So if the non-GMB is done with us, and GMB is going to be between 4 to 5 months for a 6-step synthesis, we'll be able to complete. However, if non-GMB is not done by us, it may take additional 2 more months. All right, thank you very much. I have another question for Holger, and then we'll close to the end of the webinar. But Holger, there is also a question about, so can you comment a bit, or what is special about the beamline you use?
I can comment on this. Our general idea is to produce the best possible data quality, and the beamline we use here is run by EMBL in Hamburg at the Petra 3. synchrotron, and it is known for its superior data qualities, and it's a highly flexible and highly adaptable beamline, and yeah, the Beamline scientists involved made everything possible to speed up our processes here. And in general, I would say, I mean, I can, if someone is really interested in this, I can talk about the X-ray beam profile that makes a big difference compared to all other beamlines worldwide. And there's other beamlines that might be faster on paper, but in the end, they are probably not.
Thank you, Hulger. I'm just looking, I'm cautious about the time. So I know there's many more questions in the chat and in the Q&A, which I thank you for that. We'll also follow up offline. You will have also on the last slide, maybe you can go there, the contacts. So I would encourage all of you to contact the people from the panelists. So you have the contacts, or you can also contact myself that I can facilitate this. So thank you so much for joining today's presentation. So if you have any additional questions, or you would like to get in touch with our team at Scientistcom or any of the presenters, so that information you can see on the slide. The recording of this presentation will be sent to you by email within the next day. So thank you, thanks to our speakers for their involvement in the presentation. And of course, thanks again to all of you for joining us. And we hope that you found the presentations helpful. So thank you very much to all of you.
Thank you.